So hopefully a little bit of a simpler slide. It's pictures rather than a lot of words and arrows, but another important slide that's going to allow us to think about the differences between what happens when we have need state, let's say low blood sugar, and fed state, high blood sugar. Okay, so three sort of things I'm going to talk about in this slide with the overarching umbrella of what's different between muscle and liver cells is we're going to talk about the transporters that are different on the surfaces of the glucose transporters that are different between liver cells and muscle cells. We're going to talk about the differences and how they respond to signals, extracellular cell signals. And then we're going to talk about the differences between what may be present or not present on the inside of these cells that sort of allows them to sort of respond differently to these situations. So big picture first here, might be a little bit hard to see with colors, but hopefully you'll see that there's two different kinds of arrows that we have, two different colors of arrows that we have here. Any blue arrows are need state. And remember, when we're talking about need state, that's glucose or energy. And then we're going to have fed state. And those are going to be in red. Okay? So first, let's talk about the differences that we have here in our glucose transporters. Okay? So I want to highlight that over here in liver cells, we have GLUT2 transporters. So we've got GLUT2 transporters. These are always present. And remember that any glucose transporter operates in a facilitated diffusion fashion, so we're always going to be moving glucose from wherever it's higher into wherever it's lower. So the liver has a glucose transporter that's always present, okay? And what helps sort of regulate whether or not it's actually pulling in glucose is we've talked about this, I believe, before. Okay, there's going to be another video that's on this, talking about the importance of the hexokinase activity that we have within our liver versus our muscle cells, okay? So what that really means is that we're going to have a liver GLUT2 receptor that's not going to be pulling in glucose to the extent that it would create a state of hypoglycemia. Okay, and that has to do with, remember, once you pull that glucose in, if you're going to use it in the first steps of glycolysis, hexokinase is the first step. So there's a different hexokinase that's in liver cells that has a much higher Km value, meaning it's got to have really high concentrations of glucose in order to be pulling in glucose, doing that conversion to G6P, and then allowing more glucose to be brought in. So it sort of makes sure that the liver cell doesn't put us into a state of hypoglycemia. Okay, so those are the GLUT2 transporters that we see on liver cells. Let's contrast that to what we see here with our GLUT4 transporters. And our GLUT4 transporters are what we see on our muscle cells. There's other cells that have GLUT4 transporters, but the key with GLUT4 transporters is that they are insulin sensitive. So we're going to have our GLUT4 transporters. These are insulin sensitive, meaning that they are only going to be present when you have high blood sugar. So only present with high levels of glucose. Okay, and specifically, we're just looking at these two cell types, okay, but a great way to contrast GLUT2 receptors, which are always present, insulin insensitive, always present, but because of biochemistry that happens sort of downstream of pulling glucose in, the GLUT2 receptors that are on the liver are not going to cause a state of hypoglycemia. GLUT4 receptors are only present when there's high insulin, which means there's high blood sugar. So again, that is going to allow us to pull that blood glucose into our muscle cells. Okay, so let's talk next about what we have here with these four signals, four extracellular signals. I'm just going to label what they all are here, and then we'll talk about them one at a time. And we'll talk about whether or not muscle cells versus liver cells specifically respond to them. So we're going to talk first about epinephrine. Okay, so epinephrine is these blue guys that we have here. So we've got our little blue guys here, epinephrine. So there's our epinephrine. So epinephrine is going to be those blue guys. And what we're going to see is that both liver and muscle respond to epinephrine. But remember what epinephrine really is. Epinephrine is our fight or flight sort of hormone. And so it kind of makes sense that if something's going on where we are stimulated that we're going to have to fight or flee, it makes sense that the liver should be breaking down glycogen and flooding the blood with glucose so that we have it as an energy source. But it also makes sense that we're going to be doing the same thing in liver cells because we want those liver cells to be able to have the energy that they need. Said liver, those muscle cells, that we're going to want those muscle cells to be able to have the energy that they need to do their movement and have us fight or flee, whatever it is that we happen to need to do. Let's contrast that right now to glucagon. Remember, these are both need state signals, but if we look at glucagon here, and glucagon is going to be sort of that yellowish guy here. Here's glucagon. Glucagon secreted by the pancreas, okay? Only liver is going to respond to this. Only liver is going to respond to this. And this is really important because only liver cells can help remedy a low blood sugar level. So again, glucagon is only going to be secreted when blood sugar levels fall. You can have sort of normal to even a little bit elevated blood sugar levels, but you can have epinephrine be secreted by your adrenal glands, okay? And in that case, both cells will respond to that, but only the liver is going to respond to low blood sugar levels. And we're going to highlight in just a second why that is when we kind of put this third piece together. So these are both need state signals, okay? So I'm going to highlight that these are both need state signals, but again, differentially responding depending on what cell type you are. Now, when we think about glucose and insulin, again, these are both fed state signals. So let's go ahead and just highlight where we see these. So again, we're going to have, here's our glucose, okay, little green guys here. And then our insulin is red there. Okay, so insulin, again, secreted from the pancreas. Glucose, again, just we've got high blood sugar because we've eaten. Okay, so these are both need state signals. So highlighting that these are need state signals. Okay, but we are going to have both liver and muscle respond to high blood sugar levels, and that's because they're going to help pull it in as a way to help remedy that high blood sugar level. We talked about the importance of having sufficient muscle mass because muscles serve as a great glucose sink to kind of maintain glucose homeostasis. Your liver does as well, right? But it's great to, you can't modulate the amount of liver that you really have, but you really can modulate the amount of skeletal muscle you have. So another important reason for having that. So we're going to have both are going to respond to and pull in glucose. And again, that's key for homeostasis. Okay? And so the third piece to sort of put on here is to highlight another reason. We know that muscles do not respond to low blood sugar levels. We know that they can't help if we've dropped below that 5 millimolar level. So there's sort of three reasons why they don't. Okay? So this is again why muscle is sort of local use only. That glycogen that is in muscle cells is only for their use. And there's three reasons why that's going to be the case. Well, the first is they have no glucagon receptors. Right, so if we highlight where those glucagon receptors are, change colors here, those glucagon receptors, right? We see them here on the liver cell. They're absent on muscle cells. So they don't even hear the signal to begin with. But beyond that, there's two other reasons that they cannot help. They do not have any G6Pase enzyme. So let's take a second here and look over at what happens in the liver cell, right? If we are responding to glucagon, right? We've got other videos here that go through the details that happen in this with these arrows, right? We're going to activate adenylate cyclase to increase cyclic AMP. Cyclic AMP is going to initiate the pKa phosphorylation cascade that ultimately breaks down glycogen into G6P, but then we are going to have G6Pase. That's a critical component to being able to generate free glucose. Because remember, only free glucose can be exported from across transporters and exported into the bloodstream. So we need to have G6Pase to get in to have those free glucose, okay? And then the other situation, the other reason why muscle cells cannot do that is they only have GLUT4. Okay, and so basically when we are in a need state, those GLUT4 receptors are going to be absent from the cell surface. So highlighting again this cascade from glucagon receptor, okay, which is missing in muscle cells. They are also missing G6Pase, so they can't even make it into free glucose, that even if there was a transporter there, GLUT2 is always present on liver. The GLUT4 transporter is not even going to be present if we are in a low blood sugar level. So 3 reasons why we are not going to be able to have muscle cells remedy a low blood sugar level. So that's why for glucose homeostasis, muscles are really good for helping to remedy high blood sugar, but they're not going to be able to help with low blood sugar. So just to wrap up this video, let's just make sure to look at the picture now that we have all of these details around with what's happening, and let's just take some of these arrows and walk through need state versus fed state and address what happens within each sort of cell type here. So if we're talking about the pancreas secreting glucagon, that must mean that we are in a glucose need state. We highlighted there's nothing happening over here with muscle cells, but we can see that glucagon binds to glucagon receptors on the liver cell. We already went through this, activates the adenylate cyclase, increases cyclic AMP levels. We're going to activate pKa to undergo that phosphorylation cascade to ultimately activate phosphorylase, which is going to break down glycogen to G6P. G6Pase is going to break it down into free glucose. Because these GLUT2 transporters are always present, we're going to be able to take the high concentration of glucose that's now in liver cells, export it out to the bloodstream, which again is low blood sugar, and remedy that low blood sugar state. Wow, I know there's a lot here. All right, let's talk about the other sort of need state situation. Maybe we need some energy, right? Maybe we need to be doing some fighting or fleeing, right? Epinephrine is going to be an important signal for that. Adrenal glands secrete epinephrine. We're going to have receptors on both liver and muscle cells. I'm not going to get into the weeds right now, but the difference between these alpha and beta adrenergic receptors, we will note that liver has both, and that helps to not only increase cyclic AMP levels, but also cause an influx of calcium. Both of those are going to facilitate glycogen degradation in the liver. Epinephrine is going to have a similar response in muscle cells in terms of the cyclic AMP cascade with those beta adrenergic receptors, going to cause the breakdown of glycogen into G6P, highlighting what's happening here. When you have G6P enter glycolysis rather than glucose, you actually get extra ATP out because you're bypassing that first hexokinase step that would make G6P. So we actually get a boost sort of in the amount of ATP we're going to get when we have the glycolysis happening in liver cells with glucose derived from glycogen, because it's going to be G6P. Last guy, let's talk about fed state. Okay, fed state, pancreas is going to be secreting insulin in response to high blood sugar levels. Now, we'll get into the weeds a little bit later on as to why there's differing numbers of insulin receptors on muscle versus liver, but this is an important piece. Okay, but whether it's insulin response on muscle or on liver cells, the same thing basically is going to happen. Again, we're going to have that insulin stimulated protein kinase that's going to activate PP1. PP1 is that phosphatase that puts us in that D-phospho system. It's going to allow for glycogen synthesis, upregulating the enzyme synthase. So whether it's in the muscle cells or it's in the liver cells, you're going to have active transporters. Remember, high blood sugar means that both GLUT2 and GLUT4 transporters are present. High blood sugar outside of the cell is going to be pulled in both situations, and we're going to store that as glycogen. Remembering, though, glycogen stores are limited, both in skeletal muscle and in liver. So once we've tapped out those reserves, then the liver or the muscle cells aren't going to be able to continue to take in glucose, but the liver will, because those GLUT2 transporters are still functioning. And not shown on this slide, but excess glucose ultimately will be taken through glycolysis, made into acetyl-CoA, and then we're going to shunt that to the synthesis of fatty acids and triglycerides for storage in a reservoir that does not have a limited capacity, right? And that is our adipocytes. Okay, lots that here, but hopefully with pictures and a little bit more of a high level view rather than our other video that was much more in the weeds of the phosphorylation or dephosphorylation cascades, helping us to understand what happens in high blood sugar versus low blood sugar.